Okay, part 2 of this week's session is looking at how protein is exported from the Golgi and the endoplasmic reticulum, and looking at all of the possible places where proteins can go when exported in the vesicle bound system. So you've seen this slide before. We've just heard about how endocytosis occurs using clathrin coated pits, and that brings material into the endosomes and to the lysosomes. Any material that goes from the Golgi to the lysosomes also tends to go in clathrin coated pits. But we're going to go through all the possible routes of protein transport, starting from the ER, going through to the Golgi stack maturation, looking at the retrieval pathway from the Golgi back to the endoplasmic reticulum. So this is shown here. So this is Golgi's transport out of, ER to Golgi transport out of the ER into the Golgi, and this is in a COP2 coated vesicle. We're then going to look at Golgi stack maturation. We're then going to look at how material gets back to the endoplasmic reticulum. Typically receptors that take material forwards at point 1 need to get back at point 3. So we're going to look at some of those key receptors and the signal peptides on those. We'll look at the Golgi stack itself and how proteins get through that Golgi stack and what happens to them en route. And in particular, we'll focus on transport to the lysosome. So how do those hydrolytic enzymes get through the Golgi stack into the lysosomes without eating away the Golgi and the endosomes in the process? So this is a one-way traffic system. And then finally, we'll look at secretion of proteins out of the cell. So firstly, we're going to focus on this point. We've got proteins in the endoplasmic reticulum. How do they get to the Golgi? So this slide shows how proteins get out of the Golgi. And as we heard about last week, proteins get secreted into the endoplasmic reticulum, and they'll be lacking in secondary and tertiary structure. They then get refolded by chaperone proteins in the endoplasmic reticulum, and once they're correctly folded, they can be dispatched from the endoplasmic reticulum. So here are some misfolded proteins that have been bound up to receptors. We can ignore those. The proteins that want to leave the endoplasmic reticulum will have a specific signal sequence upon them that will bind to a specific receptor, which will be congregating on some exit sites from the endoplasmic reticulum. So this is much like the endocytosis, but in reverse. We have got receptors binding to proteins that are destined to be exported. Specific signal sequences on there will bind to these specific receptors here. These receptors are then bound into some adapter proteins and with COP2. COP2 is that vesicle coat that we find on vesicles that are leaving the endoplasmic reticulum and heading to the Golgi. So we get a congregation of proteins for export at these exit sites, where we'll get a congregation of receptors, which will also recruit in quite a lot of COP2. Then we'll get budding of this vesicle, and that will go off to the Golgi. Now what you need to have in the back of your mind is how do these receptors get back, because we need to recycle them. And there are also ER resident proteins that should hang around in the endoplasmic reticulum, such as some of the chaperone proteins. And if they accidentally get transported forwards, not bound to a receptor, but just get transported forwards, they need to be brought back to the endoplasmic reticulum, because that's where they do their job. So those are going to be brought back in the endoplasmic reticulum retrieval pathway, which we'll look at point 3 on our diagram. So the second main point on this diagram is going to be how do these structures form here. Before the Golgi stack, we find these structures called vesicular tubular clusters. So we're going to look at those next. So before we get to the full Golgi stacks, we have this structure called the vesicular tubular cluster. And a vesicular tubular cluster is basically a congregation of transport vesicles which have come away from the endoplasmic reticulum in COP2 coated vesicles. These then uncoat to be more of a naked transport vesicle, and then these vesicles congregate and fuse to form the vesicular tubular clusters. And as we saw last week about vesicle fusion, we have T snares and V snares, T snares and V snares sort of stick together almost like Velcro. They can be separated by an enzyme called NSF once they've been fused, and then they can be separated again to different parts of a vesicle. So we've got a couple of vesicles heading this way. They've both got T snares and V snares. They can come together. The T and V snares can link. That can cause fusion, and we end up with these large vesicular tubular clusters, which are clusters of transport vesicles that have recently come from the endoplasmic reticulum. Now what we can see on this diagram also is a retrieval pathway of material going from here in a COP1 coated vesicle back to the endoplasmic reticulum, and that will be coming up in the next few slides. So this is all the retrieval pathway is also always in COP1 coated vesicles. So we're up to point 3 on the diagram. We've had material go from the ER to the vesicular tubular clusters, and then subsequently they'll go to the Golgi stack, but we also at this point will explain how material gets back to the ER on the ER retrieval pathway. So here's a diagram of a protein wanting to move forward. So in sort of the gold squares are some secretory proteins, and those secretory proteins will be bound up with some receptors which will recognize them. They'll have a specific exit signal peptide on them that will bind to receptors. So those receptors, some receptors will be carrying this secretory protein forwards, and that will work its way to the vesicular tubular clusters, subsequently onto the Golgi stack, and as it passes through the Golgi stack, that may well become modified, but as time goes on, that protein will become concentrated in the Trans Golgi Network, where the proteins will be dispatched for secretion or wherever they are destined to go. At the same time, endoplasmic reticulum resident proteins, such as the chaperone proteins, but other proteins that just naturally reside in the endoplasmic reticulum, they will also sometimes travel forwards, not bound to receptors, but they will find themselves travelling forwards, and that would be wasteful to the cell, so those need to be retrieved back. So the way those are retrieved back are by specific receptors which are actually deliberately sent forward just to return those proteins. So at this point here, this is an exit site in a COP2 vesicle. There's a receptor there. It's not actually binding this secretory protein, something else will have bound that. This is purely going forwards so that when it gets to the Golgi, it can mop up all of this ER resident protein and bring it back to the ER. You know, this could be a chaperone protein, it could be a protein that's involved in helping to stabilize proteins as they go through the Golgi stack, but it needs to be brought back. So this is what we call the endoplasmic reticulum retrieval pathway. And this is shown in more detail here. This receptor, which is bringing back these ER resident proteins, are called K-del receptors. K-del is just the 4 amino acids, lysine and so on, which that is a signal peptide on these proteins. So this is a K-del receptor. This binds to any protein with an exposed K-del signal sequence. This will bind to this. When the K-del protein with a K-del on it binds to a K-del receptor, that will recruit the COP1 coat, and the COP1 coat will encourage vesicle formation from the Golgi, and that will be retrieved back to the endoplasmic reticulum. So this is the amino acid sequence of K-del, that little sequence there, and all of those soluble ER proteins tend to have that sequence expressed on them so they can be brought back. Now the K-del receptor itself interacts with COP2 via a lysine-lysine XX, and where X can be any amino acid, but it's a run of 4 amino acids that's crucial, but it's the first 2 lysines that are really crucial. So this KKXX sequence is what's linking the K-del receptor to the COP1 protein itself. So that's another protein transport sequence that you need to know. I think I mentioned there's only a few signal sequences that you really need to know. One of them's K-del, one of them's this KKXX, and the other one is the nuclear import sequence that we're going to cover next week. And this is just showing the KKXX interaction with the COP1 code there. Okay, so we've got our proteins out of the ER to the vesicular tubular clusters. We've got our ER resident proteins and receptors back to the endoplasmic reticulum itself. So what we're now going to cover is the Golgi stack itself and how proteins move forwards through the Golgi stack. Now there are two proposed models of how things move forward through the Golgi stack, from the Cis Golgi Network through the Golgi stacks into the Trans Golgi Network, and one of them is the vesicular transport model, where vesicles bud off and bind to the next Golgi stack and then bud off and go to the next Golgi stack and repeat the process. So that's one thing that you can see happening. And if you do live studies using proteins that are tagged with green fluorescent protein, you can observe proteins doing this, and that is a relatively quick process. On a much more long-term timescale, there's this cisternal maturation model, which says that rather than vesicles budding off and joining the next stack, that the whole stacks slowly move in this direction and slowly mature. So the Cis Golgi Network becomes the first Golgi stack, and that becomes the next one, then the next one, and so on. And there's evidence to show when you start tagging proteins with fluorescent molecules that both of these can happen simultaneously. Proteins can go forwards within vesicles, whilst at the same time the stacks can move slowly forwards over a longer time period and mature. Now what we're going to cover in the next section, which will be part 5, is the sorts of post-translational modifications that occur to proteins as they pass through this Golgi stack, whether it's passing through vesicles in this way or whether it's the whole stack maturing, doesn't really matter. But what we're going to cover next is how lysosomal targeted proteins go through, and you'll just see one example of a post-translational modification that gets put on in the Golgi apparatus itself. So we're going to be looking specifically at the example of targeting proteins to the endosomes and specifically the lysosomes, and we're going to look at a specific tag, which is a mannose-6-phosphate tag, but in doing so we'll look at how a protein passes through the Golgi stacks and at different stages has different post-translational modifications attached, removed and then modified to create the final active protein. So just as a reminder, we mentioned this in the last session, but lysosomes are these membrane-filled compartments that are filled with hydrolytic enzymes. They're called lysosomal hydrolasers. They're acidic-loving enzymes, and they will break down virtually anything. These are synthesized into the endoplasmic reticulum as proenzymes and inactive enzymes, and then as they pass through the Golgi apparatus, they are tagged with post-translational modifications. So these are modifications to the amino acids, and they are tagged on in a very specific way to change the protein's behavior. These lysosomal hydrolasers are tagged with mannose-6-phosphate, and this is added on in several stages, which I'll show you on the next slide. Once the mannose-6-phosphate tag has been applied to that protein, the mannose-6-phosphate receptors are used to bind those hydrolasers and interact with clathrin at this point here. So to put this into context, the protein has been synthesized into the endoplasmic reticulum here. It's been transported through the Golgi stack, and in going through the stack, the mannose-6-phosphate tag has been added. Once that tag is fully formed, it can bind to mannose-6-phosphate receptors, which will be congregating here. Those mannose-6-phosphate receptors will interact with clathrin, cause a vesicle to bud off, and that will then head towards the late endosome, and then the late endosome will mature into an endolysosome and finally into a lysosome. So the first thing that happens is the protein is received from the endoplasmic reticulum, from a COP2 coated vesicle. It will go through the vesicular tubular clusters and it will receive a tag of mannose. Mannose is just a carbohydrate. This mannose-bound protein is then passed on to the next Golgi stack, where an N-acetyl-glucosamine phosphate tag is added, which is this one here. And then After that, the protein moves further through the Golgi stack to the next compartment, where just the N-acetyl glucosamine is cleaved off to reveal a mannose 6-phosphate signal here. So next step is binding of the mannose 6-phosphate tag to the mannose 6-phosphate receptor, and once that occurs, that will recruit clathrin. That will form a clathrin-coated vesicle, causing that to bud off and create a naked transport vesicle because the clathrin will get recycled back. This transport vesicle will then head towards the early endosomes, which are slightly more acidic than the transport vesicles. That acidity is driven by ATP-driven acid pumps, as we saw in the lysosome slides previously. Under these conditions, that will cause dissociation from the mannose 6-phosphate receptor, which will then go back, be recycled back to the Trans Golgi Network, and this receptor can be constantly recycled. That leaves the mannose 6-phosphate bound acid hydrolase here. We then get cleavage of the phosphate group off, and we get the lysosomal hydrolase precursor here. And this low pH of the endosomes results in this being a unidirectional transport system, so you can't get this enzyme being sent back to this part. So once it's detached from the receptor, there's no way of this enzyme getting transporting backwards towards the Golgi. Now at this point, this lysosomal hydrolase precursor will be dispatched in a vesicle with a appropriate RAB tag, and we covered RABs last week, so there'll be a specific RAB for targeting to the lysosomes, and so this protein will then be dispatched in a vesicle with that particular tag, that tag will bind to the RAB effectors that we talked about last week, and then we'll get vesicle fusion and then release of the lysosomal hydrolase precursor enzyme into the lysosome. Now this is just one specific example of a post-translational modification, and for secreted proteins, many proteins are post-translationally modified, so they're secreted into the ER, they pass through the Golgi stack, they can be phosphorylated, sulfated, have carbohydrate molecules on them, particularly extracellular matrix proteins will have things like glycosaminoglycans added to them as they pass through here. So this is not all post-translational modifications, this is just one specific example of one specific protein. And as you look at more proteins, you'll find a whole diverse array of post-translational modifications that get added in the Golgi usually to activate a protein, but there are lots of different types. Okay, we're on to the final stretch of this session, and we're now on to secretion or exocytosis. So our protein's gone from the ER into the Golgi, passed through the Golgi. Will it be modified in some way? Maybe something like antibodies that go through there and they're glycosylated, and light chains and heavy chains that are connected together. A lot of that post-translational modification occurs going through the Golgi stack, and then at some point that protein needs to be either secreted or sent to the plasma membrane if it's a receptor. So proteins leave the Trans Golgi Network and then they are sent for secretion to the plasma membrane, and this is what we call exocytosis. And there are two secretory pathways, what we call constitutive and regulated. So constitutive is providing proteins and lipids for the membrane, sort of rebuilding them. You have to remember that for every vesicle that is taken up by endocytosis, that bit of plasma membrane needs to be rebuilt. So a vesicle needs to go to the plasma membrane to rebuild that bit of plasma membrane. Also, membrane proteins that are normally expressed, your LDL receptors, need to be sent there in the first place, and secretion of secretory proteins and particular extracellular matrix proteins are released. And we call this the constitutive pathway because cells just generally chug out receptor to the membrane, rebuild the membrane and release some extracellular matrix. So it's relatively non-selective, it just happens continuously to maintain that cell's environment. In contrast, we've got the regulated secretory system, and this is a specialist secretion system for things like hormones, neurotransmitters, digestive enzymes, that type of thing. This can be incredibly quick. So if you think about neurotransmitters, you've got, this could be a synapse, you've got a signal binding to a receptor, and that signal binds to a receptor and then immediately causes the release of something from a vesicle that is stored just below the plasma membrane. So in terms of neurotransmission, that happens in a very small fraction of a second. In terms of hormone release, that can occur very quickly as well. Just think about how quickly adrenaline is released. And then sometimes this can happen at a slightly slower, more sedate pace for some secretory hormones. But these all come under the regulated, because the release of the vesicle is reliant upon some sort of signal from outside the cell, something binding to a receptor, triggering the release of those vesicles that can happen almost instantaneously or be a little bit slower in terms of many hormone secretions. So we just call that the regulated pathway rather than the constitutive pathway. So in this regulated pathway, proteins for secretion are really concentrated and stored in secretory vesicles just below the plasma membrane, and then the vesicles bind to the plasma membrane when they're told to do so by some sort of signalling here. So this is a summary of all of the places that material is likely to go from the Trans Golgi Network. We've covered mannose-6-phosphate mediated signalling to the lysosome. We've got signal mediated secretion for regulated secretion, and we have got what we call the constitutive secretory pathway, also sometimes referred to as the default pathway. And this default pathway is your secretion of extracellular matrix, you rebuilding the plasma membrane and sending receptors to the cell surface. And this is just a list of all of the things that we've covered in this video and the previous video, looking at also linking back to things that we learnt about last week, linking back to the RABs effectors, snares I mentioned there, vesicle fusion. We've got some of the key signal peptides, the KDEL receptors, the KKXX sequence for retrieval, back to the ER. We've got these details on the various maturation models of the Golgi stack, details on post-translational modifications, the lysosomal hydrolyses going through to the lysosomes, and then finally we have the three main destinations from the Golgi stack, being constitutive secretion, regulated secretion, or sent to the lysosomes. Okay, so that's the end of vesicle transport of proteins throughout the cell. Next week's sessions are going to cover nuclear import and export in detail, so looking at the signal sequences and see what they interact with, and we'll also cover mitochondrial import. Mitochondrial import is not that dissimilar from endoplasmic reticulum post-translational import, which we covered last week.